About introduction to codon optimization. Codon optimization improves the efficiency of protein synthesis by adapting gene sequence to host preferred codons. Organisms exhibit codon bias, favoring certain synonym codons, optimized codons, enhance translational efficiency and yield. And the purpose of codon optimization is improve recombinant protein expression and gene performance. And I also elaborate this introduction of codon optimization with the diagram in which I elaborate the recombinant proteins are made through codon optimization and other process. I think you understand with this picture. Codon usage and bias. Codon usage bias unequal frequency of synonyms. Codon usage across different organisms means we use unequal frequency codons and on different organisms. Understand driven by transfer RNA abundance and translation efficiency. For example, E. coli favors GCG for alanine, whereas humans may prefer GCC. Means these are codons, and mostly E. coli favor codon, and humans prefer GCC codon. And I also elaborate codon usage bias with this flow diagram. In this diagram, I elaborate codon usage bias in a circular way, and I think you will get to know about that. And then we will discuss what is codon optimization table. A table identifying optimal codon for each amino acids in a target organism. Component amino acids, preferred codon, usage of frequency. What's the purpose of optimization table? Guides gene design for higher expression in a host. And I also give the example of a table in which I discuss the amino acid and their optimal codon usage frequency, and alanine GCG optimal codon and its usage frequency is 31.5%, and so on. All of these amino acids have their optimal codon and their usage frequency. Why is codon optimization important? What's the benefit, and what's their application? First of all, we discuss about the benefits. It's increase the protein yield in recombinant expression system, reduce cellular energy burden during translation, improve expression in heterologous hosts, means bacteria, yeast, mammals. Applications: recombinant protein production, gene therapy and vaccine development, synthetic biology for metabolic engineering. I also elaborate a flow diagram in which a codon optimization and non-codon optimization are compared, and a flowchart will describe about the non-codon and codon optimization table. And then we will discuss about recombinant protein production and their importance in our biology. Optimization foreign genes for expression in hosts like E. coli, RCHO cells, biopharmaceuticals, synthetic biology, and gene therapy. All of these are mainly applications in which we discuss about codon optimization, and we use codon optimization, and it will be very beneficial in biopharmaceuticals because we improve vaccine and antibody production. Synthetic biology, we build efficient genetic circuits and metabolic pathway. And then gene therapy, it will be very beneficial because in modern age gene therapy is very important in our life, in human life, and in our animals. We also use this and test that. Then we will discuss visualization of codon usage bias. I elaborate this with the heat map and graph showing synonyms codon frequency in various organisms. For example, codon preference comparison between E. coli and humans. I elaborate that with the flowchart, or I mean it's a diagram in which different colors elaborate the visualization of codon usage. Then I will summarize about that topic. Codon optimization is essential for enhancing translation efficiency. Codon optimization tables are vital tools for designing genes suited to host organisms and application in biotechnology for synthetic and therapeutic. And then all of that. Thanks for your time. Have a good day, sir. And all of that.